we are going to continue with the cell division today, okay?
Mhm.
All right. So till today, whatever I have taught, is there anything you need to clarify? Any doubts?
No.
No, all clear? Yes.
Mhm.
Shall we continue?
Yeah.
Yeah. Okay. Perfect. All right. So we'll continue with cell division. Just a recap of what we have done on cell division in the last class. Do you remember this? I will be sharing you the window now, and you can see.
Yeah.
See this?
Mhm.
Yes. Okay. Do you remember this? Yes.
Yeah.
We had the cell cycle, right? Every cell will undergo a phase which is called as cell cycle. Okay. All right. Cell cycle. And can you see the G1 phase, the G2 phase, and S phase, M phase? Yes.
Yeah.
Yes. Can you tell me what happens in the S phase?
Um, DNA replication.
Yes, DNA replication. You see there, see? DNA replication, duplication of DNA, right? Yes. And G1 phase. Can you tell me what happens in the G1 phase?
Protein synthesis.
Yes, protein synthesis. Centriole, see? Centriole duplication completed in G2 phase. And can you tell me what happens in the M phase?
Cell division.
Yes, the actual cell division, right?
Mhm.
Yeah. So we are going to study now what is happening in the cell division. So let us start here. Okay. So cell division. So we have two types of cell division. Okay. If we talk about any kind of cell division, we definitely have two types of cell division. And the two types of cell division are, one is mitosis and another one is meiosis. Okay, mitosis and meiosis. So mitosis, so we are going to study more on mitosis. Okay,
Mhm.
mitosis and just a little bit in meiosis so that you get a clear picture of what you are studying. Mitosis, this is a type of cell division which is termed as equational 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 cell division. That means, for example, if I have a cell here, this cell will equally divide to form two daughter cell. This is the mother cell. This is the daughter cell. Okay, so whatever the mother cell, whatever contents are there in the mother cell, that is equally divided among the two daughter cell. Okay,
Mhm.
suppose, for example, if the mother cell has chromosome, okay, it has its chromosome number is what? 46. Then each of the daughters will also have 46. It remains the same, does not change. Is this clear?
Yeah.
Yeah. Okay. The other cell which we call as meiosis, this is called as reductional reductional cell reductional cell division. Now reductional means, if I have a cell here and when at the at the end it forms two daughter cells, all right, okay, if this is 46, each will be half of what is 46, 23 plus 23. Clear?
Yeah.
Yes. So I'm not going to go further into meiosis here. We are going to go more into the mitosis. Okay, just know that it is called reductional division, and it takes place when? During the process of gametogenesis. That means during the formation of gamete. Gamete formation. Only then in our body the meiosis takes place. But every time there is, if cells have to be replaced in our body, if cells have to be formed in our body, then it's definitely mitosis. It's definitely the equational division. For example, if you cut your hand, a part of your skin is gone, right? So after some day, when you put ointment, and after some day, what you observe, a new skin develops. Okay, so how is that made possible? That is made possible because of mitosis. Okay,
Mhm.
mitosis will always increase the number of cells. We find mitosis taking place in the plants. Okay, in the plants, when you cut a plant, okay, suppose there's a plant, and then you just take off the leaf. After sometimes, what do you see? Suppose, for example, if you have a plant here, and here one leaf is growing, just cut off this. After sometimes, you'll see in the same place small leaf buds are developing. Do you see?
Yeah.
Yes, that is also because of mitosis. In our skin, also mitosis. So that means, if I have to say, mitosis is taking place in all the cells of our body except the gamete cells. Okay, in all the cells of the body, mitosis will take place except the place where gametes are formed. Okay,
Yeah.
except during the formation of gamete, the remaining are all mitosis. Okay, even in when the gametes are formed initially, mitosis takes place, but the main division of gamete formation is meiosis. Is this point clear, Sana?
Yeah.
Yeah. Okay, let us go back to our cell cycle here. Now, what we observed in the last class was, each time a cell passes from one phase to the other, there are some kind of enzymes controlling their movement from G1 phase to S phase, S phase to G2, G2 to M, and then from G0 phase again to G1 phase. Okay,
Mhm.
so these enzymes, I'll write it here. Okay, so these enzymes are nothing but what we call, see, for example, we have a cell cycle. Let me write here, cell cycle. Okay, cell cycle. So in cell cycle, we have G1 phase, we have S phase, we have G2 and M phase. So each time a cell goes from one phase to the other, this movement of the cell is controlled by enzymes called CDK, cyclin, or what we call as cyclin dependent kinase, cyclin dependent kinase. Is this clear?
Yeah.
Yeah. Okay, cyclin dependent kinase. And one more thing, Sana, from next class onwards, I will also try to solve, some questions, because there are some questions, like, for example, just want to know, I want that you should know a little bit of the magnification, okay, the calculation. Yes. So that without me uploading the question paper, I'll not be able to show you, because we need images for that, right? Yes. Yeah, and then we need the scale also, right? So I will be, I will be solving every day, not only theory part, but also I will be solving questions. Okay, all right, that be fine. Yes.
Yeah.
Yeah. Okay, you look tired.
Yeah.
Came back from school.
Mhm.
Yes.
Yeah.
Okay, so shall we start? Yes.
Yeah.
Okay. All right. So cyclin dependent kinase. These kinases are, okay, these kinases, let us see. These kinases are specific enzymes, specific compounds that control, okay, that will control the movement, that will control the movement of cell from one part, or on one phase to the other. Without their controlling, it is not possible for the cell to function at all. Okay, so we have cyclins, okay, cyclin dependent kinase. So unless and until, suppose for G1 phase to S phase, so we have G1 phase cyclin. So for every phase there is a separate cyclin, and if the cyclin is not functioning, then the cell cannot move from one phase to the other. Is this point clear?
Yeah.
Yeah. Okay. All right, so let's go to the process of mitosis. So I told you that mitosis, M phase, equational division. Okay, so mitosis is divided into many phases. Okay, so first phase, Sana, do you remember interphase?
Yeah.
So interphase is just the preparatory phase. So we're not going to talk. In the last class, we have said about interphase. So today we are just going to concentrate on the M phase, that is the mitosis. Okay, mitosis will have the first phase, which is called as prophase. Then it has metaphase. Okay, after metaphase, anaphase, and then we have the telophase. Okay, prophase, metaphase, anaphase, telophase. Okay, so let us see here what happens in the prophase. Let us go one by one. So okay, sorry, one second. After that, all right, then we have cytokinesis. Now this prophase to telophase is called as karyokinesis. That means division of nucleus. Cytokinesis is division of cytoplasm. Cytoplasm. Did you understand these two, Sana? Yes?
Mhm.
Okay. Now, next, we'll see. Let's see here. So, we have the first phase. Okay, so we're going to go into the first phase. Just a second. We're going to go into the first phase and see what are the properties of the first phase. So the first phase is called as prophase. This is the first phase of mitosis. Okay, first phase of mitosis. See, usually in the interphase chromosome, the interphase stage, the nucleus appears very large. Okay, now see here. All right, I'll just give you a small information about one more organelle that will be found. Okay, so now this is the cell. Okay, inside the cell we have is the nucleus. Okay, all right, inside the cell we have nucleus. Okay, so the nucleus will be as it is there. All right, now, just have a look at the chromosomes in the interphase stage. This is the cell in the interphase. This is the nucleus in the interphase. When you look into the chromosomes, this is the chromosome. This is what we call as chromatin. Chromatin fibers. This is interphase. During the interphase, this is how the cell appears. But we are coming to the prophase, which is the first phase of cell division. Now you will find the nucleus. You will also find, okay, you will also find one. Let us see here. You will find that the chromosomes have start to condense. Okay, the chromosomes have start to condense means they are becoming thicker. So I'll write here point number one. The chromosomes, or instead of chromosomes we'll write, okay, chromatin fibers start to condense. Okay, condense, that means they become thicker. They become thicker. Okay, they become thicker to form what? To form chromosomes. They become thicker to form chromosomes. See the difference. In the interphase state, this is how it becomes. In the prophase stage, this is how it becomes. Okay.
Mhm.
So condensed. So we are seeing that it is coming to the shape of the chromosome. Now, we will also find one structure in the cell just adjacent to the nucleus. I've mentioned about this. Okay, when we're doing the cell structure, this is called as the centriole. Okay, this is called as what? Centriole. Or this entire structure, let me just write whatever we have done. This is called as the centrosome. And the perpendicular structures that you find here too, these are centrioles. Centrioles are present only in animal cell. Only in animal cell they are present.
Mhm.
Plant cells do not have centrioles. Please remember, plant cells are devoid of centrioles. Okay, centrioles, centrioles are microtubular structures. Microtubular structures. Okay, that means they are made up of protein. Okay, they're made up of protein. Okay, that protein is called as microtubule. Is this clear?
Yeah.
Okay, they are made up of protein called as microtubule. Okay, they're made up of protein, which is called as what? Microtubule. Okay, any doubt here?
No.
Okay, all right, so centrioles. Okay, so okay, so this centriole, what happens? One of the centriole will remain here, whereas the other will start to shift to the opposite pole. Okay, one centriole will remain here. One centriole will remain here. One will remain here, whereas the other one starts to move towards opposite poles. Is this point clear?
Yeah.
Yes. Okay. So, what is the function of centriole here? The function of centriole is to form spindle fibers. Function of centriole is to form spindle fibers. Clear?
Yeah.
Yeah. Okay. So let's see what is the spindle fibers. Okay. So this is in the prophase. Now, what happens in the prophase, we will also see, the nucleolus will slowly, you'll also have a structure here. Let me just write here this structure. Okay, so this is the nucleus. This is the nucleolus. This is the centriole. All right, and these are the condensed condensed chromosome.
Mhm.
Okay, condensed chromosome. Okay, next, let us see here now. Okay, now listen to this very nicely. Okay, see. Now what happens? So we are seeing the centrioles moving to the opposite pole. So this diagram which I have shown you. This is in the initial phase of prophase. As we come to the late, okay, prophase late prophase, let us see what happens here. All right, late prophase. Okay, so we already had the nucleus here, we had the nucleolus. So what happens? The nucleus begins to disintegrate. Nucleolus also begins to disintegrate. So in the late prophase, we will observe? We will observe that nucleus plus nucleus nucleolus disintegrate. Okay, nucleus plus nucleolus will what? Disintegrate. Okay, now microtubules will grow from structures like, microtubular organizing centers will be there. Okay, that means now the spindle fiber starts to form. So we will have one centriole here, another centriole reached its position. Total disintegration of the nuclear membrane will be observed, and all the chromosomes will come to lie at the equator, at the center. Can you see this? Right? Yes, right in
Yeah.
this position. All of them, all the chromosomes. So nucleus and nucleolus disintegrate. The chromosomes, okay, lie at the equatorial equatorial plane. Okay, now as the chromosomes, this is called equatorial plane where the chromosomes are there. So the centrioles will send out microtubules. Just now I read. Okay, microtubules, or in other words, we call them as spindle fibers. Okay, so now the centriole will give out what? It will give out microtubules, or we call it as spindle fibers. Spindle fibers are nothing but microtubules, and also these are made up of what? Protein. This is made up of protein, which, and the name of the protein is tubulin protein. Tubulin protein make up the microtubules or the spindle fibers. Okay, and as the spindle fiber gets attached to the chromosome, this late prophase, okay, it transition take place from late prophase. The cell is now in metaphase. Is this point clear?
Yeah.
Yes. Okay, all right. Metaphase. Now, metaphase chromosomes. Okay, so in metaphase stage, okay, metaphase stage, this will happen in the late prophase. These structures take place in the late prophase. Okay, now we will see now from late prophase, it have come to what? Metaphase. So now let us see what are the changes the cell goes during metaphase. All chromosomes all chromosomes lie at the equatorial plate. That means this region where the chromosomes are present, this is called as the equatorial plate, like the equator. Okay, the equator of the earth, the middle section, the section that runs through the middle of the globe, equator. So equatorial plates, all chromosomes lie at what? Equatorial plates. Point number 2. Next, chromosomes chromosomes are highly condensed. Condensed means they are clearly visible, right?
Mm-hmm.
Yes, they are clearly visible. Okay, they are clearly visible. All right, then now see here. So, what you observe in the chromosomes, Sana, see here. Now, if you see the chromosomes in a very clear manner, okay, very clear manner, let us see here. Okay, so now Listen to this very nicely. See, in the equatorial plate, see, all of them are lying at the center. Can you see this?
Yeah.
Yes, this is called as the mitotic spindle. Okay, it is called as what? Mitotic spindle. Clear?
Yeah.
Now the nuclear membrane, nucleolus have disappeared. They all have disappeared, and the chromosomes are quite spread. They are not confined within the limitations of the nucleus. They are quite spread. Okay, and now what we have in the mitotic spindle, we will have the chromosome. See,
Yeah.
okay. Now let us see the structure of the chromosome, how they are attached. So suppose, for example, if I see the chromosome, if I see the chromosome, this is how the chromosomes would be. One arm of the chromosome. This is the second arm. Okay. All right. Now, these two are what? They are arms of the chromosome and called as chromatids. Okay, what they're called as
Yeah.
chromatids. This structure at the center, this structure at the center, this is called as centromere or primary constriction. Okay, primary constriction. There can be some cells or some chromosomes which may have a second constriction, and this would be, can you tell me, Sana, what this can be? Any idea?
Mm,
Yes, anything which you have studied, recall? You studied, right? Yes.
yeah.
Yes, what is it called? This is called as secondary constriction. Okay, and this chromosome, here inside this chromosome is our highly coiled DNA. Okay, this is our
Yeah.
highly coiled DNA. Okay, now you see here in this section, in this bulge region, the DNA will be coiled, and it will contain repeated sequence or repeated DNA segments. Lot of repetitions will be there and highly coiled. So this section is called as, this is called as SATELLIT, satellite. And the chromosomes that have satellite are called as sat chromosomes. Okay,
Mm-hmm.
satellite chromosomes. All chromosomes don't have, only few chromosomes have the satellite chromosome. Okay, all right. Now, the arms are called as what? Chromatids. This primary constriction is called as centromere. Now, in this primary constriction, there are two knob or button-like structure. These knob or button-like structure, this is called as kinetochore. Kinetochore. Okay, now you see here, Sana, how you see the diagram, and I'm going to draw the chromosomes and you'll understand how they are attached. So I'm going to take one chromosome which is there in the equatorial plate. Okay, okay. So as I said, we have here kinetochore. Now, see very nicely. Spindle fiber from one pole will come and attach to this kinetochore. Spindle fiber from the other pole will come and attach to this kinetochore.
Mm-hmm.
You understand? Yes?
Yeah.
Yes. So this is how the chromosomes will be arranged in the metaphase stage.
Mm-hmm.
Okay, in the metaphase stage. Now you see here. The arms, if you can see, the arms are facing towards the poles. whereas the centromere is at the equatorial plate. Arms all are facing, all the arms are directed towards, all the arms are directed towards the opposite poles. Okay, all of them are directed towards the opposite poles. Clear? Yes.
Yeah.
But okay, they're directed towards the opposite pole, but the centromere is at the center, at the equatorial plate. This is the characteristic of metaphase chromosome. Okay, so all right, the chromatids, chromatids or arms are directing towards, okay, are directing towards the poles, whereas the centromere is at the equatorial plate. Is this clear?
Yeah.
Yes, equatorial plate. Okay, then next, let's see here. Okay, equatorial plate. So this is what it is going to be, right? Okay, so now if I have to study, if suppose for example I want to study the structure of chromosomes, okay, now which will be the best stage for me to study the structure of chromosome? The answer is I can study the structure of chromosome best in the metaphase stage.
Mm-hmm.
Okay, in which stage? In the metaphase stage. Okay, so I can, okay, so chromosome structure is best studied in metaphase stage. Why? Because, okay, the structure is clearly visible. Is this point clear?
Yeah.
Ah, it is clearly visible. Clear or not? Yes. This
Mm-hmm.
is the metaphase. Okay, this is the metaphase. Now, what happens? Now, let us see here. Now, we are going to go into the anaphase. We are going to go into anaphase. So, what happens is that now we have our chromosomes in the equatorial plane, or what we call as the mitotic spindle. We also have the centrioles attached to each of the kinetochore. This is the metaphase chromosome. Now, a little drastic change takes place during the anaphase. Now, you see, the spindle fibers are attached to the kinetochore. So, they will start pulling the chromosomes to the opposite pole. Okay, they start pulling the chromosomes to the opposite pole. One second. Yeah. Now, can you see this? Can you see the page?
Yeah.
Yeah. Now you see this chromosome that I've said. So when what happens during anaphase, okay. Now this spindle fiber which is there, it will start to pull the chromosome towards opposite pole. Okay, the spindle fibers will pull the chromosome to opposite pole. As a result, the chromosome will divide. What divides? The centromere divides. Okay, what divides? Centromere divides. Clear or not?
Yeah.
Yes, centromere divides. Okay, centromere divides and the two chromosomes move towards opposite poles. This starts the beginning of what we call as anaphase. Okay, now we'll see here. Okay, let's see here. All right. Now you see here. The centromere has divided. See, this is how the chromosomes will be. Can you see this, Sana?
Yeah.
See? Haven't they divided at the centromere? Yes?
Yeah.
Yes. So in anaphase, the centromere divides. Then the chromosomes are pulled towards opposite pole. Like this, they'll be pulled towards opposite pole. Now, what is the position, Sana? Here I gave you a position. I said here chromatids are chromatids directing towards the poles, centromere and towards the equatorial plate. Now look at this. Look at this. It's exactly opposite of what is happening. See the arms are facing towards the equatorial plate, whereas the centromere is facing towards the pole.
Mm-hmm.
So here centromere facing towards the pole, and arms or chromatids directed towards the center. This point is clear.
Yeah.
Yes. Got it or not? Yes.
Yeah.
Okay. All right. So now you see, if only the chromosomes are being pulled, they have a tendency to move away. We don't want the chromosomes to simply move away, from their position. Okay. So we will need something to stabilize them. So now let us study the different spindle fibers. So this diagram, can you see clearly, Sana?
Yeah.
Yes. So can you see the purple ones? Yes.
Mm-hmm.
Yes. The spindle fibers, can you tell me they are attached to which part of the chromosome?
Uh, the centrum?
Centromere. Yes or no?
Yeah.
Yes. So the spindle fiber which is attached to the centromere, this is called as discontinuous. One second. Discontinuous fibers which are attached to the centromere. Clear? Yes,
Yeah.
this is attached to centromere. Okay, I'll use a second color. There is one more which will be running directly from one pole to the other. Can you see this?
Yeah.
Yes. This green color, this is called as continuous fibers or spindle fibers that appears from one pole to the other. The third one will be present between the centriole. Sorry, between the sorry between the centromere of one chromosome and the centromere of the other chromosome. Can you see the Sana, the blue color?
Mm-hmm.
Yes. These are called as interzonal fibers, and they join from what? One centromere to the other, right? One
Yeah.
centromere to the other. These are the three types of the spindle fibers that we will see during cell division. As the cell division progresses, as the cell division progresses, the chromosomes moves further and further away. Okay, so we have seen how the structure will look like now.
Yeah.
Okay, all right. So now what happens? You can observe that the chromosomes starts to move towards opposite poles, and as they reach the opposite poles, they appear somewhat like this. Okay, under the microscope, this is what you're going to observe. Do you see the images which is there in the book?
Yeah.
Okay, page number 53. All right, you can see the images. All right, see how microscopic images of anaphase looks like, and how the the other structures, the diagrams look like. Do you have the book with you?
I do.
Yeah, can you take out page number 53?
Yeah.
Yes, can you see?
Yeah.
Yeah. Okay, so see the microscopic images. Okay, this is how the microscopic image of anaphase will look like. The chromosomes are absolutely on the opposite poles. Okay, on the opposite poles. The daughter chromosomes have separated. Okay, they have separated. Now, finally, what happens now? Okay, so you can see this anaphase. See, this is anaphase.
Mhm.
Anaphase or late anaphase, we can see. Okay, and you can observe, right? Some of the fibers may be visible like this. All right. Okay. Then now, what happens as the chromosome returns to the opposite pole? Now, what happens? The chromosome slowly starts to decondense. Okay, the chromosomes will slowly start to decondense, not fully, but in a very gradual state. They will start to decondense like this. Okay,
Mhm.
all right. So, this is the phase that we call as telophase. Okay, in telophase, the chromosomes, chromosomes starts to what? Decondense and acquire the shape of fibers. Okay, slowly, we can also observe the nuclear membrane reappearing. Nuclear membrane reappearing at each pole. Nucleolus also reappearing. We will also see, okay, so I'll write here reappearance of nuclear membrane and nucleolus. Nuclear membrane and nucleolus. Clear?
Yeah.
Yes. Okay. So these are the two things that you need to observe. This is called as what? Telophase. So by the time it is late telophase, we will see that the nuclear membrane have. All right, me just take another one, no problem. So this is telophase. This is late telophase. We will observe the reappearance of a complete nuclear membrane, nucleolus, and the chromosomes have totally decondensed. Okay, and they have formed again spindle fibers. So we can see nucleus, nuclear membrane, we can see nucleolus, And we can also observe chromatin network or chromatin fibers. But can you tell me, Sana, is the cell division complete?
No.
No. What is required?
The cytoplasm to divide?
Yes, the cytoplasm to divide. We need cytokinesis. So, cytokinesis. Okay, now, all right. So, let's work on cytokinesis and okay, perfect. We'll work on cytokinesis. Cytokinesis. That is division of the cytoplasm. So, we see here, okay, the nuclear membrane, nucleolus. So now why I have drawn two shapes, can you tell me?
Um,
Yes, this is,
Animal and plant?
yes, this is animal cell. This is plant cell. Okay, animal cell and plant cell. Now, next. So now what we have observed, we have observed fully condensed chromosomes. See? All right. Now it's the time of cytokinesis. So if it's an animal cell, if it's an animal cell, right from, okay, if it's an animal cell, this is called as the cell membrane. Okay. If it's a plant cell, this is called as the cell wall. So if it's an animal cell, so what happens? A furrow formation takes place. See, furrow formation. This furrow formation will deepen and it will proceed towards the center. Then finally, okay, it will divide the entire cell. So in animal, what happens? Furrow formation occurs and which goes towards the center. Okay, finally, after the completion of cytokinesis, finally, we will then get two complete cells. Clear?
Yeah.
Yeah. Two complete cells. This is two complete cells after the process of what? Cytokinesis. See? Two complete.
Mm-hmm.
This is animal cell. Next, the plant cell. In the plant cell cytokinesis, right at the center, there will be, okay, phragmoplast deposition or formation. Phragmoplast formation or deposition. Okay, phragmoplast. You know what is phragmoplast?
No.
Yes, pragmoplast. Any idea?
No.
No, not heard. Yes. You know Golgi complex?
Yeah.
Yes, Golgi complex. Okay, Golgi complex will, disintegrate and form small, small components or fragments. Okay, fragments. This is called as phragmoplast. This is formed from where? The Golgi complex. Okay, Golgi complex. Now, this phragmoplast, what it will do is that it will extend towards the cell wall. See the difference. Animal cell extending towards the center, plant cell extending towards the cell wall. Okay, cell wall, and finally forming a plate like structure which is called as the cell plate. This plate will deepen and then finally the cell wall will form. In between the cell walls will have middle lamella and then the two cells will divide. So at the end we will get two plant cells. Okay, all right. This is vacuole. Inside this, we will have the chromosomes. It's clear, Sana? Yes.
Yeah.
Yeah. Understood. Yes. Cytokinesis.
Yeah.
See? Right. Yes. Okay. When we study the process of cell division, we study something called as mitotic index.
Mm-hmm.
Okay. So we use this mitotic index just to check out. And what is mitotic index? It is the number of cells in the mitosis divided by the total number of cells. That means, for example, if you are observing in a microscope, so you should understand that in particular specimen that you've taken, how many cells are actually undergoing mitosis, and how many are in a state of no division. Okay, in a state of no division. Is this clear? Yes.
Mm-hmm.
Yeah. So number of cells by number of total number of cells in mitosis by total number of cells. Okay, so it is the ratio, it is ratio, and it can be, we can estimate it. Okay, in this particular cell at this time, this is the mitotic index. That means this is the percentage, or this is the amount of how many cells are actually undergoing division. Okay, so number of cells by mitosis. Okay, got it or not? Yes.
Yeah.
Yeah. Now we understand mitotics. Mitotic index helps us to understand the rate of cell division. Okay, the rate of cell division, that means how faster the cell is dividing or not, that we will understand using mitotic spindle. Suppose, for example, in any kind of disease, okay, in disease, some of the diseases like cancer, the cell division is continuous and it is very fast, not like normal cells. So in order to estimate how fast the tumor is growing, how fast the cancer is spreading, we should know how fast the tumor cell is developing. Yes, this point
Yeah.
is clear, Sana? Yes.
Yeah.
Any doubt?
No.
No. Okay. So what happens is that we have, we have specific cells in our body. Okay, specific normal cells, and these are dividing normally. Usually what happens, in our body, if we have one cell, after mitosis, Sana, how many cell we will get?
Two.
Two cells. So okay, so this will undergo differentiation, and this will continue the process of what? Division. Similarly, this will undergo differentiation. This will continue the process of division. Okay. So what is happening here? What is happening here? Let's just see. Okay, this is normal cell. But in our cell, okay, this is our normal cell. I'll write here. This is our normal cell. Now, due to any kind of mutagen is cancer causing agent. It can be, okay, mutagen can be radiation. When you are exposed to radiation, mutagen can be certain chemicals. Okay, mutagen can be certain chemicals. It can be, okay, mutagen can be certain chemicals. Mutagen also can be, some kind of gases, or x-rays, or any let's see, radiation, chemicals, yes, some kind of gases. Okay, what they do is that, now in our cell, okay, in our cell there is a gene. Okay, in our cell there is a gene which is called as what? Which is called as proto oncogene. These mutagens, what they will do is that they will change this proto oncogenes, change this proto oncogene which is functioning normally. Okay, to what? To oncogene. And when oncogene is formed, then what happens to the cell? This cell will undergo excessive proliferation. Proliferation means continuous and excessive cell division. This point is clear, Sana?
Yeah.
Yeah, any doubt here?
No.
Okay. Now, excessive proliferation. So, what happens here? So, ultraviolet rays, radiations, chemical radiations, all these, what will do? They'll change our genes. So, now let us see what happens. Okay, all right. So, now suppose now this proto-oncogene has it has mutated our cell. Okay, it has become oncogene. This is mutation only, right? So this, suppose this cell is mutated, so this cell is undergoing excessive uncontrolled cell division. Can you see this?
Yeah.
Yes, this is nothing but what it is. It is uncontrolled uncontrolled cell division. Okay, uncontrolled cell division. So, and this is what? This is covered by a small by a membrane. Okay, and inside this membrane the cells are continuously dividing. All right, and it is forming a tumor. Okay, and this tumor is called as benign tumor. What is it called? Benign
Yeah.
tumor. Now, this tumor is so many cells. So, does it need food, Sana, to survive?
Oh, yeah.
Yes. So, what they will do? They will sometimes get the food from other cell, or they may even direct the blood vessel like this. Okay. They may direct the blood vessel. But benign tumor, okay, so they are causing what? Metastatic growth. We call it as metastatic growth, new growth. Okay, metastatic growth in one organ. This is okay. So, one second. Yes. All right. So, they're causing here growth. Okay, tumor growth. Metastasis, metastatic growth. When we talk about tumor, when we talk about cancer, we use the word metastasis. Have you heard?
No.
Yes, metastasis. Okay, so here, now see here. So this tumor is confined to a region. This is confined, it is not spreading. Okay, so this is called as what tumor? It is called as benign tumor. This tumor is confined to a region. This is not spreading, so it does not cause cancer. Is it clear?
Yeah.
Okay. Another of this mutated cell undergoing excessive cell division, uncontrolled cell division. Now here there is no membrane, and the blood vessel is directed up to them. See, can you see this? Yes.
Yeah.
So this cancer cells, what they do, they use the blood vessel to go to some other organ. This is organ number 1. And they go to some other organ number 2. Okay, organ number 2.
Mhm.
And there they form growth. They grow secondary metastasis. Neoplastic growth, growth of tumor, neoplastic growth, neoplastic growth, spreading of tumor, spreading, metastasis. Okay, now this tumor is called as malignant tumor, and it can spread. Benign tumor do not spread. It remains confined to a region. Is this clear? Yes.
Yes.
So malignant tumor will go to another organ and show secondary growth. That means the cancer is spreading to next organ. There are various causes of cancer: smoking, chewing tobacco, getting exposed to high levels of radiation, so many things. Okay, is this clear? Yes.
Yeah.
Yeah. All right. So this is all about cell division. Okay, this is all about cell division. Now, there are few more things I want to say. Okay, so here now you see here, when we say cell division, okay, when we are talking, see, we spoke about cell division, how the cells will divide. Now cancer cell will also divide in the same way: mitosis, metastasis, anaphase, everything will be there. But what is the important thing, that cancerous cells are dividing at a very faster rate.
Mm-hmm.
Very fast rate they will divide. Okay, at a very fast rate they will divide. Is this point clear? Yes.
Yeah.
Any doubt?
No.
No, all clear? Yes.
Yeah.
Yes, Sana. Yes. Can you see your book? And if you have any doubt, can you tell me if I can explain you few more things which you have not understood? Because I too have the same book, so I need some clarification from you so that I can understand whether you've understood or not.
No, not that I see.
Yes, okay. I think we have done everything right.
Oh, there's one thing. It's like a cycling graph.
Sorry, which one? Which one? Which page?
Page 56.
Page
56.
56. Okay, yes. Cyclines, yes. Just now I have done
Yeah,
cycling.
like the graph.
Yes, yes, the graph. Okay, all right. Come to the graph. Let's come to the graph. Okay, all right. Now, see here. What they're saying is that the concentration of cyclins, okay, cyclins in various phases of mitosis. Can you see the G1, S, G2 mitosis phase? Mitosis, as you can write it, is the M phase.
Mm-hmm.
Okay, mitosis, which is given there, it's the M phase. So that can be either meiosis or it can be mitosis. Clear? So
Yeah.
this here we are doing mitosis. Okay. Now, see here. What they have given, the pink color, can you see?
Yeah.
Yeah. Okay, the pink color that is given is called as cyclin D. Okay, and what it does is that, it triggers. I told you that unless and until one cyclin is activated, the cell cannot go from one phase to the other phase. No.
Mm-hmm.
Can you see what is written there? Cyclin D triggers cells to move from GO to G1.
Yeah.
Yeah. Now, what is that GO?
G-O.
See here, see here. I'm showing you something. GO. You'll come to know. Yeah. Now, can you see?
Oh, yeah.
Yes. What is GO?
Um.
G not phase.
Mm-hmm.
I told you that after the cell division is completed, the cell can go into a stage where it is not active. Okay, so suppose the body requires cell, then the cell has to go from G naught to first phase. Can you see on the whiteboard G1, right?
Yeah.
I've given an arrow there, right? So it says the cyclin D, the pink color, okay, the pink color band that you can see. All right, the pink color band that you can see, cyclin D, it triggers the cell to move from, okay, to trigger the cell to move from G naught to G1. In the book, there is written GO, right? So you can write it there as G naught.
Mm-hmm.
G naught, you can write there G naught to G1, and from G1 into S phase. Can you see this?
Yeah.
Yes. So that means cycling D is active, means now the body requires cell. So the cell will move from G0 to G1. It will carry out all protein synthesis, everything. It's ready now. Now the cyclin D will push the cell to S phase. And I have
Okay.
told you that once a cell reaches S phase, anyhow, it has to divide because
Mhm.
a cell cannot remain with duplicated chromosome. The next one, cyclin E prepares the cell for DNA for replication in S phase. Now the cell is in S phase. And what is the most important function of S phase? Cell replication, DNA
Mhm.
duplication, DNA replication.
Yeah.
So some compound has to be there. With that compound will trigger the cell, trigger the DNA to undergo what? Replication. That means duplicate itself.
Mhm.
Right? Suppose, Sana, for example, okay, for example, we both have the same book, right?
Mhm.
Yes, okay. Now, suppose, for example, me and you, we need the same book. Okay, we need the same book. Now, but only one book is available. So, am I going to take half of the book and you're going to take half of the book? Will it be like that? No. So, if you have a book, I will take a scanning copy of the book and I will make a second copy of the book, right? Yes.
Mhm.
In the
Yeah.
same way, we don't just divide the DNA, we duplicate the DNA. Okay,
Mhm.
we duplicate. So that duplication of DNA is triggered by cyclin E. Okay,
Okay.
next. Next, can you see cyclin A? Can you see cyclin A?
Yeah.
Cyclin A activates DNA replication inside the nucleus in a space. Okay,
Mhm.
now cyclin E will, what it will do? It will prepare for DNA replication. So DNA replication preparation means nucleotide is required, DNA polymerase is required, enzymes are required. So cell will start to collect all those enzymes, and then cyclin A will finally activate DNA replication
Mhm.
inside the cell. That means now the DNA duplication will take place. Is this clear?
Yeah.
Yes. Okay. Why do we need cyclin E, Sana? Why do we need cyclin E? Why do we need cyclin E?
Hello.
Why do we need cyclin E? It,
Oh,
yeah.
it prepares the cell for DNA
Yeah,
replication.
but why the preparation is required? Why not directly cyclin A?
Because
Yes.
the proteins need to be synthesized.
Yes, yes, correct. You're correct. Tell me. Yes, you're correct. Yes, because we need the proteins, right?
Mhm.
Right? Yes or no?
Yeah.
We need the proteins. Without studying, can you go and write your exam? No. You need to prepare for the exam. The
Mhm.
DNA, our nucleus needs to prepare. It needs to get nucleotides. It needs to get let's see, enzymes. Only then it will be able to, carry out the process of division. So that's why E is required. Preparation. A is for active cell, active replication. Cyclin B promotes the assembly of mitotic spindle and other tasks in the cytoplasm to prepare for mitosis. That means what? Cyclin B is active only in M phase. Cyclin B is active only in which phase?
And
M
face.
phase. It is active only in M phase. Is this point clear?
Yeah.
Yes, it is active only in M phase. So what is the function? What is its function? What it will do is that, you know, preparation of mitotic spindle, preparation of the spindle fiber, separation of the chromosome during anaphase, condensation of the chromosome, decondensation of the chromosomes, okay, condensation of chromatin fibers, decondensation of chromosomes. These functions will be carried out by cyclin B.
Okay.
Yes, this,
Yeah.
be carried out by cyclin B. Is this clear? Yes?
Yeah.
Yeah. Okay. All right. So have you understood the graph? Yes?
Yeah.
Yeah. Can we just look into the graph once? Yes?
Mm-hmm.
Can you see the pink color graph is cyclin D, right?
Yeah.
So sorry, not cyclin D, sorry. Can you tell me what is the pink color band?
Cyclin D.
So, okay, S. Then let us see what is the green color for
Oh, cyclin E.
E. That is, it prepares, right? So you can
Mm-hmm.
see the peak in S phase. What about the purple?
Cyclin A.
Okay, and then the red,
Cyclin B.
cyclin B, right?
Mm-hmm.
Yes. So different kind of cyclines are there here. Okay, mitotic index I have taught you. Yes.
Yeah.
Okay. Cell theory. Are you prepared with the cell theory? Yes. What does the cell theory say?
Um, all cells come from pre-existing cells.
Yes. And
Um, they independent units.
yes, cells are the basic units and independent units of organisms. What else?
Um,
Yes.
smallest unit of life, or
Sorry.
um,
Two. Yes. The cells arise from pre-existing cells.
yeah.
Yes, cells arise from pre-existing cells. Yes,
Mm-hmm.
yes, yes. Okay. So there are some discrepancies, there are some exceptions to cell theory, right?
Mm-hmm.
Can you tell me what are the exceptions to cell theory?
There's a paramecium.
Yes, no, not paramecium, not paramecium.
Oh, no, 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 no. The algae. The
Algae, yes, acetabularia, right?
yeah.
Yes, acetabularia and
There is
striated muscles.
yeah.
Striated muscles, yes, striated muscles. The red blood cells, the red blood cells,
Mm-hmm.
okay, the red blood cells, and and can you tell me fungi, right?
Acetate fungi.
Yes, septate fungi, right? That means the hyphae is a septate hyphae. Okay. All
Mm-hmm.
right. So just let us have a look at that. Okay. So with discrepancies, when I say, so the first one, we will go to the first one, which is what? RBC or red blood corpuscles. Why? Because RBC, when we see, it is biconcave, it is enucleated. There is no nucleus, right?
Mm-hmm.
There is no nucleus. It's enucleated. All right. And can you tell me, Sana, why RBC does not have nucleus?
I'm not sure.
What is the main function of RBC?
They're red blood cells. So
What is the main function of RBC?
um,
They have something called as hemoglobin.
yeah.
Yes. So what is the function of hemoglobin?
It brings
Yes.
oxygen.
Yes, correct. It brings oxygen. So, Sana, for example, its most important function is to bring oxygen. And if it has all the organelles, nucleus, mitochondria, can it make space for more hemoglobin?
No.
No, that's the reason why it does not have most of the cell organelles. Then
Mm-hmm.
second we have is striated muscles. Striated muscles. Striated muscles. These are what? These are with light and dark bands. These are with light and dark bands. This will be the dark band, this is the light band. Then it is multinucleated, right? And it is like, it is long like fiber, it is fibrous. All right, striated muscles, which defies the law of cell theory. The third one is aseptate fungi.
Mhm.
Aseptate fungi or hyphae. In any fungi, their main body is hyphae, and we have two types of hyphae. So in this aseptate hyphae, there is no septa or no cross walls. Okay, usually septate hyphae will be like this. See.
Mhm.
Okay, so they follow the rules. New single nucleus will be there. This is called as septa. So this is septate hyphae, and it's found in the higher fungi. This is aseptate hyphae, multicellular. The cytoplasm is flowing, okay, without any obstructions. These are found in the lower fungi. And last one is acetabularia. Acetabularia is a green algae, right? Acetabularia is what algae? Green algae. What do you understand by green algae?
Um, it's like a fungi.
No.
Um,
Yes. Acetabularia. This is a green algae. A
mhm.
green algae, also called as chlorophyce. Green algae or chlorophyce. Clear?
Yeah.
Now, this is unicellular. All green algae are unicellular. But then acetabularia is a large size green algae. It looks like almost like a plant. Almost like a plant. You can see it. It's like a plant. But it is unicellular algae.
Mhm.
Unicellular in cell theory always means that it has a single cell. Okay, it has a single cell. So this is
Yeah.
unicellular, but it is large size. Clear? Yes. Yes. Okay. Any more doubts? Yes. Cell size, cell limitations I have done. Yes. That's
Mhm.
important. Okay. Then what are stem cells?
Oh, cells that can be um specialized.
Yes, cells that can be specialized to form other structures.
Yeah.
Okay, cells that can be specialized to form other structures. Totipotent cell, capable of giving rise to any cell type. Okay, any
Mhm.
cell type. So when we do tissue culture and all, we are always, playing around with totipotent cell, playing around with stem cell. Why do they say nowadays, after the child is born, preserve the umbilical cord? Why?
Mhm.
Because if the child grows up and suffers from any diseases, umbilical cord contains stem cells. Those stem cells will be helped, they will be used to regenerate the body parts or to cure the diseases. Clear?
Okay.
Yes. Bone marrow cancer, leukemia. Yes. All right. Okay. So our cell theory chapter is done. Okay. All right. In the next class, I'll just upload one paper and we are going to do on the magnification part. That will be the end of that. Okay. Is there anything else, Sana? Anything else?
I don't think so.
No. Everything has been covered, right? Yes.
Yeah.
Have you understood?
Yeah.
Yes. Okay. So, what the next chapter we will do in the next class will
Mhm.
be molecular biology, right?
Yeah.
Yes. Okay. In school, up to where you have finished molecular biology?
We're mostly done.
Okay. Almost done. Yes.
Yeah.
Yeah. Okay. Fine. So we're going to start with molecular biology in the next class. Okay, and we'll be doing some questions as well. Clear?
Okay.
Okay. So shall we end the class here?
Yeah. Okay.
Yes.
Thank
Okay.
you.
All right. Bye. Take care.
Bye.
Bye.